Okay, so I guess we'll get started then. It's my pleasure to welcome you all to the second distinguished colloquium of the semester, featuring a friend and colleague of mine, Ronald Walsworth, from the Harvard Smithsonian Center for Astrophysics. So Ron got his PhD from Harvard University in Ike Silvera's group in 1991, something like that?
Yep,
yeah.
That's right, yep.
And we overlapped kind of by seeing each other at conferences and things because Ron got involved in some work on optical pumping and polarized gases, which kind of kept him busy with things like imaging, but also, you know, over the years, he branched out into other various fields of atomic physics. And he kind of had, as we discussed before the talk, kind of an adiabatic phase transition, second order phase transition from a postdoc-y kind of person all the way to a senior lecturer at Harvard and a research appointment at the Center for Astrophysics. And so he's kind of been there ever since.
Yeah,
and
never left.
So again,
kind of sad.
It's not a bad place
It's
to
okay.
be, right? It's all right. So anyway, it's my pleasure to welcome him. He's going to talk to us today about NV centers, which is one of the most intriguing and exciting and cutting-edge areas now in sort of tabletop atomic physics, and I'm really excited to hear your talk, Ron.
Yeah, thank you. Thank you, Brian. Yes, thank you. First time that I've been here, and I'm really enjoying myself. Thank you, and I look forward to more interactions tomorrow as well, and this evening, and the main event, the cheese thing we're going to be having after I finish this talk.
Oh, I was supposed to mention that since this is a distinguished colloquium, we have a reception afterwards, and you're more than welcome to join. Afterwards, meet the speaker, ask additional questions.
And so I'll try to make this swift, and we can get to the cheese. So thank you, and I'm enjoying myself, and I want to encourage particularly the younger people, the students, as we go along. This is a colloquium level talk, right? So it's attempting to be a broad overview, but there will be some technical details. There's a whole bunch of stuff we're going to go through. And if people have questions, please ask as we go along, all right? Particularly the younger people. If something seems wrong or you don't understand, I'd rather go through less material and have you understand it, at least at the level of a colloquium, right? Than just get through all the slides. I can happily just click through the slides and we can go right to the cheese if we wanted to. Okay, there we go. Thank you. Wow, all right. Look at that. Yes, so we're going to be talking about special quantum defects in diamond called nitrogen vacancy color centers, and their utility as a kind of platform technology for sensing, primarily in magnetometry, magnetic field sensing, though they can be used for sensing some other things, which I'll briefly mention as we go along. Here's my research group, and we are at the Center for Astrophysics and also on the main campus of Harvard as part of the Department of Physics and the Center for Brain Science. Students and postdocs here, all wonderful people, and I'd be happy to go on and tell you about each of them, but I won't right now unless you want to know. What we focus on is precision measurement tool building, often inspired by advances in quantum physics and trying to build bridges to applications. Applications in basic physics, applications in science, other scientific fields, and sometimes real-world applications that can go across many fields. To make this sort of thing work, you've got to have the bridge sides meet in the middle, and they have to be lined up right. So it's often key to be thinking both about things from the measurement side, how can you measure things better, but also the application side. What are the problems you're trying to solve? And that means talking with lots of people who work in these other fields. And I've always found that moving into the edge of new fields, talking with experts there, that some of my best ideas and our best ideas happen when we're maybe halfway up the learning curve. Because you have the permission to ask ignorant, dumb questions and look at things from a new way. And that's oftentimes where some new measurement modalities that can add value can come from. So in my group today, we develop these tools and we apply them in many areas. And some I'm going to talk about in this talk, but not all of them. And although these different fields seem rather disparate, they're all connected by the passion that we all have in the group for answering these questions and addressing these topics, and also measurement and how you can measure things precisely. So really we work as one research group. The people working on astronomy topics and the people working on basic fundamental physics or on quantum information science or brain science topics, we all meet together and talk about all these things regularly. Today, however, the focus is on these quantum defects in diamond. So look at this nice pretty picture. What is it? These are a whole bunch of little diamonds, about average maybe 10 microns inside. Diamond grit of the sort that's commonly used for grinding, that we just placed several years ago under a little camera and illuminated with ultraviolet light. And you see these wonderful colors. And the different colors come from different types of defects, different proportions of defects in these diamonds. Diamonds weren't made purposely to have defects. Individual little diamonds here don't necessarily have one kind of defect and another one with a different color, another kind of defect. They've got a mishmash of different defects. But this is just to give you an example of what happens, that these defects are often optically active. They can be excited and can emit radiation. They don't always have to. They can decay non-radiatively, but many of them are optically active. And in particular, in the reddish regime, there's this particular defect known as the NV, or nitrogen vacancy color center, that we're going to be focusing on in this talk. And so to give you a primer look forward, here's a little cartoon of the carbon atoms that make up the diamond lattice. And what is this NV? The NV is a nitrogen atom that replaces one of the carbons, and then a vacancy, a missing carbon atom that's nearby. And associated with that defect, which can occur naturally, some of the diamonds out of the ground, or which we can fabricate in the laboratory, we'll talk about that a little bit, are special properties. Properties where they're optically active, they typically absorb green light and emit red, and they have electronic spin. The NVs that we're talking about here have spin s = 1. They have nuclear spin, and there's hyperfine structure, and all these complicating things, but let's just start with the basic properties. Optically active, and they have spin, and the spin state can be prepared optically and read out optically. And the NVs are largely isolated from the lattice. So they can have remarkably long electron spin coherence times approaching a millisecond at room temperature. All right? So this ability to do optically, as we say here, optically detected magnetic resonance on the electron spins gives lots of opportunity for doing fun physics and applications. Okay. Prior to about this period of 2006 and 2007, NVs and many other defects in diamond were considered primarily from a quantum physics point of view. Potentially, they're single photon sources. They might be nice components in quantum information processing systems. People are still thinking about them in that direction. But starting around 2006 and 2007, myself and one of my colleagues, a couple of us identified them as being very useful as sensors, particularly sensors of magnetic fields, but also electric fields and temperature. Now, for example, at Harvard, there are many different groups across many departments which use NVs and diamond for lots of different applications. Those of us up here were the early people thinking about their applications and the utility, and then it's spread across lots of departments. SEAS means School of Engineering and Applied Sciences, Earth and Planetary Science. CBS is Center for Brain Science. Lots of different people using them for lots of different applications. So why the interest? Because of the many applications. We're not going to try to go through all of them here. I'll touch on a few, particularly those that I've been involved with as some examples to give you an idea. The ones in red, there's commercialization underway, in certain cases well underway. And I'll mention that briefly as we go along too. Lots of different applications. Okay, so in particular, magnetometry with NVs. What's special about them? How do they compare to other magnetometers? Why are so many people using them and so interested? It's really this combination of high spatial resolution. Remember, this is atomic scale defect, right, in that diamond lattice. So you can ultimately get kind of atomic scale resolution in a robust room temperature solid together with good magnetic field sensitivity. So if you plot the magnetic field sensitivity, where this is better sensitivity going in this direction, versus spatial resolution of a sensor, or better resolution in this direction, and you compare diamond typical performance to other state-of-the-art sensors you might have heard of, you'll see that they all tend to get more sensitive with coarser resolution. You have more sensors in a larger volume adding in an ensemble way to give you more signal. Or for a squid, you have a larger loop, and a squid is a flux detector, so therefore you're able to sense smaller and smaller magnetic fields. With NVs, same thing. More NVs in a volume will give you more SNR. So all these things are going to become better sensors with coarser spatial resolution, but the NV and the diamond sensors are particularly good in this high spatial resolution regime. And when you factor in the applicability of a room temperature solid rather than a cryogenic squid or a bunch of atoms, wonderful though they are, held inside a glass cell, where you can bring the diamond in direct contact or close proximity to the signal source, you can see why they might be useful for sensing things like listed here. The magnetic field from an individual neuron when it has an action potential. The NMR signal that you get from an individual protein, or imaging a single proton or a single electron if you're that far away from them. Or magnetic fields that you find, remnant magnetic fields inside of meteorites that are bits and pieces left over from stuff that was there before the formation of the planets. All those sorts of magnetic fields versus spatial resolution are plotted on this chart, and all of them have been detected in the last few years with NVs, as I will sketch for you. In particular, as I've kind of hinted at, the ability to have proximity to your target is a big advantage. That P means proton, but it can mean any other signal source. Imagine you want to detect the magnetic field, the NMR signal from an individual proton, right? The field from a proton at a dipole moment falls off as 1 over r cubed. If you can bring your sensor up close to the signal source, the field can become quite large. Then you don't necessarily need magnetic shielding. A lot of the problems with the other state-of-the-art magnetometers, wonderful though they are, is they're so sensitive in bulk systems that you have to screen out a lot of the background noise. How about making the signals bigger so you don't need to screen out the background noise? That's one of the things proximity to the signal source can give you. Some of the other features are listed here in a long list. I put colors on to try to not make it as boring as trying to read a long list, but it's still a long list, sorry. The spectral range, what different frequencies can you sense? From static DC fields up to gigahertz, many gigahertz. The spectral resolution, as I'll emphasize, has been made for AC signals has gotten as good as about 1 millihertz. It's optical preparation and detection, as I mentioned, and I'll go through that a little bit more. Remember I haven't emphasized this, but the carbon lattice has 4 directions associated with it, and you can find the NVs along any of the 4 directions. Typically, when the NVs are grown into the diamond, they will occupy these 4 directions equally. But if you want to, you can fabricate them and only have them, let's say, along 1 or 2. But typically, we'll use all 4. Then the projections of the magnetic field along those 4 axes will allow you to reconstruct the net field magnitude along the different directions and do vector magnetometry. Operate in a wide range of conditions. It's robust and biocompatible. You can bring diamond into contact with all sorts of things. And relatively simple and easy to use apparatus. So for example, this is a state-of-the-art NV diamond system which we use for doing all sorts of applications. Different NV diamond applications need somewhat different apparatus, but a lot of it can be done with a system like this that sits on a table. It might cost, if you know what you're doing, 30, 50 grand worth of components. Okay? If you don't know what you're doing, no amount of money will help you, but it's not that. And we've written a lot of papers, but it's not that hard to make these things. Obviously, you see the Helmholtz coils because you want to control magnetic fields and things like that. And cool green light, and it looks kind of space-agey, right? Inside might be a diamond chip like this. Different experiments will use diamonds of different sorts. So don't fixate on one type of diamond in one shape. They come in many shapes and sizes, but a common one is a microscope slide-like diamond. That's a little thing that you'd hold in your hand here, and it might be like this, okay? And it might be a few millimeters in diameter and 0.5 millimeter thick. It would feel a little different to you than glass because diamond has different properties, much better thermal conductivity, and kind of interesting. You could take it, a piece of ice, if you had an ice cube, hold it in your hand. If I had here, I should start doing this in Colloquium. You can cut through the ice cube amazingly because the thermal conductivity is so good. The heat from your fingers will go through the diamond and melt the ice. It's pretty cool. But it's like a robust little microscope slide in which the NVs at a density that you can control are located somewhere in the diamond, maybe 1 NV at the surface, and then you'll put your sample of interest on this little diamond, like a microscope slide, put it inside this quantum diamond microscope, as we call it, in which you're going to use the green light to excite the NVs and the intensity of the red fluorescence to measure the magnetic fields in a way that I've said several times, but have yet to explain to you how that works. It's coming, don't worry. Okay. So now's the 2 or 3 slides on what is an NV center and how we make the measurements. This is a Colloquium talk, so it's a sketchy explanation, and I apologize for that, but I want to get to the applications, just so you know. This is the simple story. So there's that cartoon again, right? There's the NV center. I haven't emphasized this yet, but it's really the NV minus. That means the NV picks up an extra electron, and that's the energetically favorable state. By the way, also the vacancy being next to the nitrogen is energetically favorable. When you fabricate the NVs inside the diamond, either in the chemical vapor deposition process grown in while you're making the diamond in a laboratory, or by implanting nitrogen ions afterwards, you then anneal the diamond to mobilize vacancies. They move around inside the lattice, tend to find the nitrogens and form NVs, and that's energetically favorable. the process to do that. Similarly, picking up this extra electron is energetically favorable. So you get a large amount of these NV minuses, which from now on I'm just going to call NVs. Okay? And they have this electronic spin s = 1, as indicated here in these 4 crystalline axes. So it is a fluorescent point quantum defect. It's a single photon emitter. It's located energetically deep within the large indirect band gap of about 5.5 electron volts that exists within diamond. Conveniently, thank you, Mother Nature, just works out that way. Many other defects that exist don't all have these nice properties. Relatively small spin-orbit interaction, which is key for some of the energy level structure behavior that I'm about to outline for you. The long coherence times I've mentioned before. Here's that nice cartoon again. You can control your density of NVs in the growth process to be low so that you can look at one NV at a time and see that they are single photon emitters, and the photon correlation function, the G2 goes to 0 if you make the time go to 0, right, that sort of thing, and play with them individually. Or you can have very high density of NVs so that the diamond is visibly purpley almost to black, and when you hit with green it glows red. You can choose the NVs to be located inside of small nanodiamonds, which can be very small. You can fabricate nanodiamonds of various shapes. Sometimes you make them in a pillar, and you have a single NV, like this ball at the bottom here. You attach it to an atomic force microscope and scan around a sample of interest to make measurements, right? Or sometimes you're going to make a kind of nano wedge, like a Toblerone of diamond that might be several microns long and have an NV every once in a while. It sits on the sample of interest, and each NV is an individual sensor, and you can push this thing around and sense. But often you have one of these two modalities, using that bulk microscope slide-like piece of diamond, either with low densities of NVs, and you pick one and you measure it because you want to do nanoscale sensing of some sample of interest right above, where you illuminate with green and take the red light out, or many NVs in a high-density surface layer, thing we call the imager or microscope modality, in which you want to do wide field magnetic imaging with high spatial resolution, now down diffraction limited to, let's say, 0.5 micron or so. Okay, and the sample goes on top, and actually most commonly we illuminate with the green light in a TIRF, total internal reflection mode, so it doesn't go into the sample, and the red fluorescence is weak and comes out as imaged onto a camera. So many of the results I'm going to show you as we move forward are going to be images, 2D images of patterns of magnetic fields from which you learn a lot of things. And it's most commonly, it's that right-hand mode, this microscope mode, this imager mode. And if that doesn't make sense to anybody, please, qualitatively, please let me know, okay? Because we're going I just want to emphasize this kind of microscope mode or this imager mode is very common. So how's the sensing performed? Let's look at the energy level structure, the energy levels that matter for the NV, and it's shown here. You have the ground electronic states, the spin states, and you have the electronically excited states, much like an atom, but of course it's inside the diamond lattice. And so both for the ground states and the excited states, there is a vibronic band on the order of 100 nanometers in wavelength. So the energy levels can be moved around by the thermal phonons inside the lattice, but those vibrations affect the spins very little. So the energy levels, if you will, shifting around randomly, but the spin states are not affected by that sort of thing. There's also some metastable states indicated here. So these energy levels, these for the s = 1, that means you have an ms = 0 and an ms = plus and minus 1 levels as shown here. And they're split, the ms = 0 and ms = plus and minus 1 are split by about 3 gigahertz. And they're split by the magnetic dipole-dipole interaction between the two electrons, which make up the s = 1 NV. Right? NV is an electron cloud. It's got 2 electrons to first approximation. That's how you can think about it. They spatially orient themselves in this spin symmetric mode here, and you have a dipole-dipole interaction that splits the ms = 0 from the other levels by about 3 gigahertz. But then, of course, the plus and minus 1 levels will shift with a Zeeman shift if you apply any magnetic field. And that rapid shifting of the energy levels with magnetic field is the source of the magnetic field sensitivity. So ultimately, you're going to be doing some sort of spectroscopy to determine how those energy levels shift to determine everything. There are electronic, it's coupled with optical transitions, the electronically excited levels, and so you typically are going to absorb green light and radiate red into the vibronic sidebands, as shown here. So that's what you do. The NV, step 1, absorbs green and fluoresces red. It turns out, however, that the plus and minus 1 levels fluoresce less at a lower rate than the ms equals 0 level does, because they can decay by the small spin orbit through this metastable state and preferentially back down to ms equals 0. So that means, let's think about that for a second. If the NV happens to be, or part of its wave function is in ms equals 0, and you illuminate with green light, it's going to absorb the green photon and emit a red photon, and then repeat that over and over again. It's just a cycling transition. Green in, red out. And that will just, and its spin state won't change, and all it does is just fluoresce. If you're in plus or minus 1, ms equal plus or minus 1, you'll absorb a green photon, and with approximately 50% probability, you'll either fluoresce with a red photon back down to the state you started in, plus or minus 1, or you'll non-radiatively decay slowly to ms equals 0. So there's this fluorescence time about 13 nanoseconds, this radiative decay time 300 nanoseconds approximately. So in the early stages, when you turn on your green light, you're going to have a photon rate, a photon flux that's going to be different between the ms equals 0 and plus or minus 1. And over a longer time, as you get out to a microsecond or so, no matter what state you started in, you're going to end up in ms equals 0. So that's your initialization step. Hit with the green light, wait a microsecond or two, you're in ms equals 0, and you've prepared your spin state. After that, the difference in the fluorescence rates, depending upon the experimental protocol, will give you information about the magnetic fields that the system has been exposed to. And so I will illustrate this in what's known as a continuous CW ODMR, optically detected magnetic resonance spectrum, as how you measure the magnetic field. This is not the only way to measure the magnetic field with the NV. There are lots of different variations that you can do depending upon what kind of magnetic field you want to determine. But I'm trying to give you an example of one straightforward way so that you can understand the underlying ideas as to how you get magnetic field information. And all the other techniques at their core depend upon the same basic physics ideas. All right? So this is a piece of data. For a single NV, it's the intensity, the number of photons, hundreds of thousands of photons per second, counts per second, coming from this particular NV. The optical collection efficiency is not perfect. It's not a perfect experiment in many ways. It's in a magnetic field, a static magnetic field along the quantization axis of around 100 Gauss. We're going to keep that fixed in this experiment as an example. And what we're going to do is we're going to do spectroscopy as we apply near-resonant microwaves and find out how the intensity of the red photons vary as a function of the microwave frequency. So let's imagine you decide, I like 2.8 gigahertz. I'm going to be far off resonance when any of these, either the ms equals 0 to minus 1 or 0 to plus 1 transitions. I like 2.8 gigahertz. And I also like 2.81 and 2.79, and I'm like moving around in there. What you're going to find is the intensity of the red photons doesn't change with microwaves, of course, because the microwaves aren't doing anything. But as you, of course, go on to one of those spin transitions, either this transition or this transition, here and here, you have a competition between the optical pumping induced by the green photons and the decay pathways and the spin changing nature of the resonant microwaves. And so you get a sharp dip and another sharp dip in the intensity of red photons that are coming out because of this competition. The green light is trying to take everything back, the NV back to ms equals 0. The resonant microwaves are trying to drive the NV to either plus 1 or minus 1, where the fluorescence rate is lower on average. And of course, this is averaging over many runs. You're just continually illuminating and measuring and measuring. So you say, if I know what the gyromagnetic ratio is of the NV, and I can measure these positions very precisely, I can get out what that magnetic field is from that splitting. And what things matter. The larger the intensity, the better. The deeper the trough, the better the contrast with the function of magnetic field or microwaves, the better. The narrower that transition, the sharper and deeper that line, the better in terms of determining the magnetic fields. You'd like a really narrow, sharp line, and that splitting would tell you what the magnetic field is. Right? So much of the work in the background going on to optimize NVs and other defects which exist in solids is to make their performance and their behavior more ideal, to give you a larger spin state contrast so that those dips are deeper, to have the coherence times longer so those spectral lines are narrower, so that you can do this sort of spectroscopy and measure what the magnetic fields are. Now, only rarely do we go through and do this whole spectroscopy to determine what the magnetic fields are, because that's not a very time efficient thing. It's nice for a talk, and it's kind of overkill. typically what's going on, and almost all the results I'm going to be showing you as we move forward, we know what the magnetic field is approximately, and we're looking for tiny either temporal or spatial variations in the magnetic field around some value. So we might sit on the side here of a resonance and look for, as a function of either time or position or both, how the intensity of the signal is going up and down based upon slight variations in the local magnetic field. Right? We're not going through to do the whole complicated thing usually, except for demonstrations. This is for static magnetic fields, a magnetic field that's kind of just be external here, just static. There are time-varying magnetic fields, ones that are complicated broadband time-varying magnetic fields that are of interest in some experiments. Some that are narrowband magnetic fields, like fields that come from NMR signals in some experiments. You wouldn't use this technique of scanning through and doing the spectroscopy for those different examples, and we have different experimental protocols that I'll only lightly touch upon when it makes sense for measuring those different types of fields, but they all, in the end, depend upon these same physical properties that are listed here, that the fluorescence, the average fluorescence rate depends upon which spin state you're in, that you can optically prepare the spin state, and that with near-resonant microwaves, either pulsed or continuous, you can manipulate the spins. Okay? Oh, yes, I want to emphasize, E and T, that's not ET phone home, that's electric fields and temperature, do different things to the energy levels. They don't cause Zeeman shifts. They take that zero field splitting of about 3 gigahertz and change it. With temperature, it's basically expanding the lattice. And with electric fields, it's in some sense stretching the bonds internally, very small amounts, which affect the dipole-dipole interaction between the electrons slightly and give change to the zero field splitting. So the spectroscopy you would do would be different, but in the end, it's a similar sort of thing, which is the intensity of light changes as a function of electric field temperature and magnetic fields in specific ways that you can design experimental protocols for. Okay. That is very far from a thorough explanation on how NV diamonds are used in magnetometry. It's a colloquium level explanation. I'm happy to talk about it over the cheese that we all want to get to soon. But now we'll go on to some highlights and some example applications, some that have been achieved and some that we're working on. And I will try to be honest with you in terms of at least my opinion about their utility and how far along in terms of translating into other fields and actually starting to solve problems these different applications are. Some have translated very far. Some are very far from succeeding and may not end up having impact. One that's translated pretty well is something like this. To take living cells, small cells, on the surface of that diamond microscope slide-like thing, and it could be one, this shows you the outline of one cell, of magnetotactic bacterium, or populations of cells, and monitor the magnetic fields they create as they wiggle around on the surface. Notice the length scale here, a micron. Magnetotactic bacteria are amazing creatures. They evolved approximately 2 billion years ago. They synthesize a series of magnetic nanoparticles inside of them, which they line up along protein and use like a compass needle to navigate in the Earth's magnetic field. All right, they're prokaryotes. They have no cell nucleus, but they're able to solve all these problems. They also are very good physicists because the size of magnetic nanoparticles that they create are just above the blocking volume that you need from these materials, so they aren't super paramagnetic. They're also great material scientists. The size of the magnetic nanoparticles are more uniform than the best state-of-the-art human techniques to be able to create them. All right, so they've got us beat, and they don't even have a cell nucleus. Okay? And there are interesting questions. Biologists want to understand the genes that are leading to this kind of behavior, and also some of the same genes are found in higher creatures like Peter Engel's here, and they like to understand what's going on with that. You know, it's probably not so he can navigate down to the cookies. It's probably, or the cheese. It's probably something else, right? It's probably being used for iron regulation, et cetera. And seeing how in living cells the magnetic field are instantiated and being able to look at them for the first time has been helpful to the biologist in a narrow niche of biology. Another high-impact application that's transferred into other areas of science, probably most furthest, is being able to map magnetic fields with micron spatial resolution and millimeter field of view in early Earth rocks and early meteorites, which I touched on before. Slices of rock in which, with isotopic and chemical composition evidence, the scientists in those fields have strongly believed that these are recorders of the magnetic fields that existed, for example, while the Earth's geodynamo was getting started, or while the planets were being formed. In the early solar system, there was, I'm told by these people, there was a gravitational instability which led to the formation of the sun, and then a large amount of swirling plasma dust around that would later lead to the planets. These was electrically charged plasma, lots of currents, probably large magnetic fields, and probably turbulent effects coupled to the magnetic fields were key for determining where planets formed and also for torquing up the cloud, because almost all the mass ends up in the proto-sun and almost all the angular momentum in the swirling dust disk. They would like recorders and they'd like a time machine to understand what those magnetic fields were back then. And in high Curie point small iron grains that are found in some of these bits and pieces left over in our solar system, which occasionally fall to the Earth and are large enough when they fall as these meteorites that the cores of them remain cool, they can take out slices and they'd like to understand the patterns of magnetic fields inside of them. No existing technology was able to satisfy these demands until we had this quantum diamond microscope. So there's Professor Roger Fu at Harvard digging up more than 4 billion-year-old rocks in Western Australia, and then that's him and his student Jay Shaw using one of the QDMs we built for them in their laboratory now to address these sorts of problems. Like this. This is an early Earth rock. This is an image of the magnetic field pattern. This is 100 micron spatial resolution here, taken with a QDM, in which we can see the ancient part of the rock in this zircon here and study what the magnetic fields are, potentially between iron-bearing grains inside of them, and distinguish between them and secondary, much younger, uninteresting pieces of rock in this experiment to attempt to learn about the early Earth geodynamo. Other technologies you might imagine, like squids, you could apply, but the squids are cryogenic, and they do not want the rock temperature change. It will change the magnetic properties. So they have to pull the squid off the surface of the rock and have this kind of spatial resolution, which of course will average over the thing of interest with lots of other signal sources and not allow you to answer their questions. So this combination of high spatial resolution and field of view to find the things of interest or patterns of fields has made these QDMs now quite successful. The National Science Foundation has funded us, get this, in a non-peer-reviewed way. The program managers just decided to give us money to build more QDMs to give to the geoscience field because that community just wanted them, and there wasn't a program and there wasn't a company to build them, et cetera. So we're in the middle of doing that now. Okay. Clinical bioassay. That's a wide field of view image. Every time you see red and blue, that's a different circulating tumor cell that's been labeled with magnetic nanoparticles in a way that's specific only to those circulating tumor cells, and then you can spread this blood sample across the diamond slide and see them. Magnetic fields pass through blood or other tissue very easily, and they're nice markers, and they have a much lower background to find bad things like biomarkers or circulating tumor cells than doing fluorescence microscopy. It's sufficiently promising that it's being commercialized, and things are going quite well. And I should, of course, declare that I'm one of the co-founders of the company, and so I have a financial interest in that activity. I also want to note that one of the nice things about being involved in startups is some of our former students can get jobs there. We were talking to the grad students at lunch about this. Okay. Another field in which this technology is being very widely applied by many groups around the world now is in basic condensed matter physics. Working with my colleague Amir Yacobi and Philip Kim, for example, we were able recently, in a paper published recently, to map the magnetic field pattern inside of magnetic insulator multi-layer systems in which you will find chiral structures related to what are called skyrmions, and determine that this is a particular nail pattern rather than a other kind of pattern of skyrmion, and in fact, the chirality of the skyrmion varies and changes as a function of height through the material. There are lots of other applications. Hydrodynamics of electron flow in graphene, ferromagnetic resonance in magnetic insulators, being able to drive spin currents into systems, creating a spin polarization gradient and determining what the spin chemical potential is. Driving systems hard and they become spin torque oscillators, some of these spintronic applications. Studying canted antiferromagnets. So there's a wide range of applications. Many groups around the world are using that. In a very real-world application, Lockheed Martin has a very large program using diamond magnetometers for detecting things that might blow up, as well as in GPS-denied navigation, wanting to navigate submarines and drones and things like this by magnetic anomalies in the Earth's crust. And they particularly like these diamond magnetometers because of two features. in addition to being, they can package them and make them low power. One is the fact that they have this vector magnetometry capability, so they can map the 3D magnetic field vector, and they don't drift because of that, right? They don't drift over long periods of time, which gang flux skates and other things will. Often, drones, submarines, et cetera, need to operate autonomously with low power in these sort of navigational modalities for weeks or months at a time and can't recalibrate.
Ron,
Yeah?
you're saying they don't drift because they're based on...
They're based on the lattice, yes. Rather than it's based on the lattice, right? And so, in fact, we recently had a paper in which we showed that we can measure simultaneously the magnetic field, rather than sequentially, the simultaneous projections of the magnetic field along the 4 NV axes, and be able to do this kind of efficient 50 picotesla per root hertz sensing. And this is Jenny Schloss, John Barry, and Matthew Turner who did that work. Okay, moving on, more applications. Sensing NMR signals from individual proteins, doing high spectral resolution NMR for the first time at the scale of individual cells, mapping the magnetic fields that come from living animals outside of a worm here, non-invasively, being able to do single neuron magnetic sensing, the diamond sensors underneath it, like in that microscope slide mode. And some new ideas on dark matter detection, which I'll end the talk with here in a little bit. So for the rest of the talk, I'll pick out 2 of these applications, the kind of NMR applications, which are making some progress, but it's not nearly as far along as things like mapping rocks and some of the condensed matter applications or the others being commercialized, and the kind of somewhat, I would admit, somewhat crazy ideas on dark matter detection at the end, which have a small, I'd say a low probability to work, but we got it funded recently, so we'll see how it goes. Okay. So single protein NMR, this was work that Igor Levchinsky and Alex Sushkov, who's now a junior faculty member at Boston University, led primarily in the groups of Hung Kung Park and Misha Lukin at Harvard, and I helped out a little bit. In this case, they used one NV center, so it's not this microscope wide field imaging mode. It's this one single NV that's about 5 nanometers below the surface. So it's really close to the surface. And they're able to occasionally, they actually had more than one NV, but they're in different locations. One here, one over there, one over there. And then they put proteins, label them to the surface at low density. And once in a while, you'd be able to find one protein that got close enough to one NV to be able to sense the NMR signal that comes from the protein. Now, there's often a layer of protons from hydrocarbons and other stuff on the surface of the diamond, and so to distinguish the protein NMR signal from background, they either deuterated the protein or they isotopically changed the carbons in it to carbon-13, which would give a distinct NMR frequency and signal. Those are one of the many ways in which this initial experiment is just a demo and not really all that useful for solving scientific problems. They could, in the end, see either H2 or carbon-13 NMR signals, and they saw that the signal frequency shifted as expected with changing magnetic field to verify that they're really seeing those signals. And a nice cool thing, there was an aspect of this experiment to make it practical. They got about a 10x boost in SNR, and using a repetitive readout technique, which maps the measurements they make on the electron spin onto the nuclear spin inside of the NV, and taking advantage of the longer T1 time of the nuclear spin, they were able to then repeatedly read out the information. That little protocol involves real entanglement and a C0 gate, so it's a little 2-qubit quantum information processor that gave a real advantage to a real experiment, right? So that's an example of quantum information processing at least doing something, okay? Really something. 10x boost in SNR is something. If you can get a 10x boost in SNR, you'll take it, all right? However, these results have very poor spectral resolution, 25 kilohertz, 6 kilohertz, something like that, and therefore are not useful scientifically. Also, you've probably altered the protein, at least at this stage. Look, it was published in Science. It's the first time anybody's seen this single protein NMR, so let's give us a break. But the reality is it's not useful scientifically. And the spectral resolution is limited by 2 phenomena. The finite T1 time, the time it takes the NV spins to reorient, which is on the order of a few milliseconds, and the decoherence time, which could be hundreds of microseconds or something like that. Even with the best fancy correlation measurement techniques, you're limited to about 100 hertz spectral resolution due to the sensor. So the NV sensor itself could only see things down to 100 hertz at best, You might think that sounds good, but hold on. And the sample is a nanoscale sample, single protein. So it has a very large kind of kilohertz sample line width due to a variety of phenomena. For the proteins, they're stuck on the surface. They're not diffusing, but many other samples you might want to sample at the nanoscale are diffusing away from the sensor rapidly, right? There's also statistical polarization fluctuations. You have a finite number of spins there, and no matter how well you get your thermal polarization in a strong magnetic field, part in 10 to the 5 or something like that, when you're down to, in a protein, tens or hundreds of spins, the square root of the number of spins will, and then the 1 over that square root will tell you what you're dealing with in terms of statistical polarizations. You can have 10% or 10% polarization fluctuations, which will dominate over the thermal polarization and basically means you're looking at a sort of narrowband noise signal, right? And that's an inherent problem at the nanoscale. So here's the thing. Remember, if you're a chemist, you already know this, useful NMR spectroscopy to do chemical identification and analysis requires, in absolute frequency units, about hertz, 1 hertz approximately, spectral resolution. Your typical so-called J-coupling, scalar couplings, the indirect nuclear spin-spin interaction mediated by the electrons in some molecule, can be on the order of hertz or tens of hertz. Chemical shifts, which are due to either the dia or paramagnetic shielding of nuclear spins due to their local environment, scale with magnetic field, and they'll go from at 0.1 Tesla, something on the order of 10 hertz, to much larger at 10 Tesla. So the chemical shifts will split apart, but these J-couplings don't scale with magnetic field to leading order. So if you're going to do state-of-the-art NMR spectroscopy, like they can do on macroscopic samples in a big system in a chemistry lab, and you claim you want to do it at the nanoscale or the micron scale, you got to do this level of spectroscopy at the hertz level, or you're not doing anything useful, except publishing science papers. So we solved that problem, at least the spectral resolution problem, in something we had in Nature in the spring. And we did it by solving those 2 problems. Remember, the sensor limits you because of its finite lifetimes, and all these statistical polarization fluctuations and diffusion stuff at the nanoscale. So problem 1, we solved with a technique called synchronized readout, which I'll tell you about, which gives you arbitrarily good spectral resolution limited by the hardware, the clock in your experiment or something like this. This was the idea originally of David Glenn, senior postdoc in the group, and Dominic Bucher, also leader on this experiment. And problem 2, the problems at the nanoscale we got away from by going to the micron scale. It's sort of like win by giving up, right? But the micron scale is still useful. The micron scale is the scale of individual cells. Conventional NMR cannot, it's too small for conventional NMR, and there's lots of problems where people care about things at the micron scale too. At the micron scale, even in the relatively wimpy magnetic field we used in our first demo experiment of 88 millitesla due to limited equipment, where the thermal polarization of the nuclear spins is about like this, it's still greater than the statistical polarization fluctuations because now you're in a larger sample with more spins, and 1 over square root of n is smaller, right? If you have 10 to the 10 spins, 1 over square root of n might be 10 to the minus 5. If 10 to the 16 spins, 1 over square root of n is about 10 to the minus 8, which is what we have there. So get a bigger sample, and statistical polarization fluctuations go away. Diffusion, that's still going on, yep. But it takes a lot longer to diffuse over a micron scale than a nanoscale, and that can become sufficiently long that that's not a problem either, right? So this is the kind of modality. Lots of NVs, maybe 10 microns thick layer of dense NVs near the diamond surface, sample of interest on the top, kind of picoliter scale sample of a few microns, come in with a green light in a turf mode so it doesn't go into the sample, collect the red fluorescence, initially on a photodiode, just a single point measurement essentially here. Later we'll do it in an imaging mode. Sensitivity about 50 picotesla per, AC sensitivity about 50 picotesla per root hertz. Quickly on the synchronized readout technique. I love ideas that are simple, easy to implement, that change the way people do science, and then in hindsight everybody said was obvious, but weren't obvious going in. And that was David Glenn's idea on synchronized readout, right? So get this, and similar to things that are used in other fields, I just love it. You might say, well, that sounds obvious, but the whole community for years in this field was trying to figure out a way to solve this problem, and his idea solved it, okay? So what does this cartoon show? It's a cartoon. At the top is the magnetic signal you want to detect. It's a decaying sinusoid. That's not real data, that's a cartoon. It could be like a single frequency NMR signal decaying, or it could be a signal you're broadcasting with an antenna that you've decided to make decay over time, or whatever. It's supposed to represent something like an NMR signal. This cartoon only has one frequency in it. You could have multiple frequency components if you want, and the signal would kind of beat down in a way. Let's say there's multiple components in frequency which are shifted from each other by a few hertz. That's the signal we're ultimately going to detect. But in the cartoon, it's just this blue thing decaying. This is the synchronized readout sensing sequence, and these gray and green blocks, the green blocks indicate when the laser is on and when you're doing either your optical pumping, preparing the NV spin state, which we talked about a few slides ago, or you're reading out the red fluorescence, right? And these periods of time in here are the periods in which you're doing the pulse sequences to the NV spins to do AC magnetometry that I'm about to tell you about. And in the end, these are your measured fluorescence that you get. And what you get is you get a beat between the signal frequency and the frequency that you determine to be sensitive to. And I'll explain that a little bit more right now. So here, inside of one of these gray blocks, this is the pulse sequence that we'd apply. And so now I'm revealing to you the reality of the slightly more complicated microwave pulse sequences we actually use if we want to do AC magnetometry. This is beginning to become serious. All the stuff that I've told you so far about how we make measurements and stuff is the kind of simple story you give in Colloquia. Now, I'm revealing some of the actual way you do these things. It's in the papers. You do things like this. You start with a pi/2 pulse. After this green preparation where you go into the ms equals 0, you take a pi/2 pulse, which tips the NVs from ms equals 0 into a coherent superposition of ms equals 0 and let's say ms equal -1. It could be plus 1, but you choose -1, right? Your resonant pulse. Then you turn off that microwave, and then you're going to start applying a series of pi pulses that are synchronized, boom, 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 to be near at a rate you're going to call a frequency you're going to call f0. All right? That's the frequency of cycling from this way to this way and back. That frequency is f0. That's nearby, but not exactly at the signal frequency. And what happens is that the blue shown here is the magnitude going up and down above 0 of the signal field projected along the NV axis. And if you apply this pi pulse sequence, a dynamic decoupling sequence, you will net add up phase to this coherent superposition of the states of the NVs. If you didn't apply the pi pulses, you say, I don't need those pi pulses. I'm going to just tip it down, and I'm going to let the AC signal, NMR signal, go along the longitudinal axis of the NVs. The first positive lobe would start to shift the phase of the NV. Then the negative lobe would shift it back. And you'd go B, 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 and it would add to 0. Because there's as many up lobes as there are down lobes. So instead, if you synchronize the flipping of the NV with what's going on, you can boom, 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 and it starts adding up more and more phase. I'm sorry, if I take your eye out. I'm at Harvard, we got a lot of money. Sorry. This is the downside of the condensed matter pointer. Okay, does that make sense? You're making the pi pulses go in near synchronicity with the AC signal to be able to get a net adding of phase. Now that's what everyone was doing. That's not the synchronized readout yet. That's the standard AC magnetometry. The synchronized readout is that what people had done before is they'd make one such measurement, and then they'd do it again and again and again, where each measurement and when you began the measurement and ended it was independent of the last one, as if you didn't know anything. The idea is you now do a series of these where you're controlling when this signal starts and the order of these, but with a clock. So that, yes, there's going to be a little bit of information loss when you're not reading out, but the beginning of the next sequence is synchronized with the ending of the last one, so that the accumulation of the information will be in phase overall. So here, it's hard to see, but there's a light gray dashed line. That's showing the sensing of by the NVs, and look, it's almost exactly in phase at this point with the signal. But if you go later, now the magnitude of the signal has decayed, and because the two frequencies are not exactly the same, you're now out of phase largely. And there's a beating between the two. And so since the NMR signals will typically last for hundreds of milliseconds or seconds, because the coherence times of the proton spins can approach a second, you can now be limited when you do NMR, not by your sensor, which you have the synchronization to do everything in phase, but by the decay of the signal. that you want to sense, which is the best you can do. If you have an even more coherent signal, like something you create from an antenna with longer coherence time, you can just average and average and average and take longer sequences, and that's where this, like, we went out to a little better than a millihertz. That's like 1,000 seconds or more, and then you get tired and stop doing it. But you could have kept going, right? So this is a wonderful, simple idea. It's easy to implement. All it means is just implement a different pulse sequence and lock it and have everything controlled with a clock and synchronized. When you do it for the first time with any technology, you can now measure chemical shifts and J couplings with the correct values with like Hertz scale spectral resolution, initially in simple organic solvents, at the micron scale. And it works easily. There are many things we're thinking about in terms of applications. I highlight single cell metabolomics. It sounds cool, metabolomics. It means monitoring metabolism and with NMR on non-invasively inside of cells and tissues with this high spatial resolution and spectral resolution, and many other things. But there's a lot of challenges. That nice data that was in the Nature paper took several hours of signal averaging. We need it much faster than that. We also need to be able to have the sensitivity so that we can go down not to 100% concentration xylene, but the actual low concentrations of millimolar and micromolar that you find in real substances of interest, and absolute, not concentration now, but absolute number sensitivity that people care about down to the femtomole range. So that's our challenges, and in recent months we've made progress by applying hyperpolarization techniques. This is now on the archive and being reviewed at Journal, whereby now we can get about a factor of 200x SNR boost and maintain the Hertz spectral resolution. So the red is the hyperpolarization technique, and the blue is without it, where you have 10,000 signal averages. It works very easily, so that now if you look at our number limit of detection, the smallest number of spins we can detect, and our sample volume, our Nature paper was here, and now we're down to here. And these are all the other previous results using microcoils and a state-of-the-art NMR on the smallest length scales. So now we can really get down in both parameters to be far better than other technologies. We still have a long way to go to be able to make a practical tool, but our dream is this one of an on-chip, single-cell NMR and MRI, doing individual cells and other samples at that length scale, and integrated in a way that could be eventually commercialized and useful. And the Moore Foundation is backing us very handsomely for going in that direction. Thank you very much. Okay, almost done. Cheese is probably being set out now. They're chilling the fine wines, whatever it is that they provide here. Maybe it's water because it's a state university, I don't know. We'll find out soon. And so we'll end with the crazy idea of maybe using defects in diamond for dark matter detection. So this is something where we're talking about directly detecting WIMP dark matter, weakly interacting massive particles. You know, are there WIMPs in the universe? They haven't discovered whether there are or not. I did not make up these acronyms. Does anyone know what the other competing, back in the 90s, the other competing model was? If you don't want me to tell that story? Machos, yes, which stands for, I didn't make this up, I'm merely reporting facts. It's massively compact halo objects. And in fact, the current director of the Center for Astrophysics, a man named Charles Alcock, who's a member of the National Academy, made his reputation as leading the macho project when he was at Lawrence Livermore and finding there are no machos. So, or just not enough machos. They actually found a few. There are a few machos, but not enough, but no WIMPs yet. Okay. All right, that's the jokes. So now it's our idea. It was, it's fun to get together with people you don't normally hang out with and ask them what the heck they care about, and then they ask you what maybe tools you have that might solve the problem. So there was some meeting at Berkeley a couple years ago, and high energy types and AMO quantum types hung out together, and we came up with this idea. But first, the background. As you might know and hear periodically, and maybe some people in your department work on, dark matter WIMP particles have not been detected, and they elude very large multi-bazillion-dollar programs, including things like liquid xenon and whatnot, to try to find these particles. It's currently thought that if one is going to go forward and detect WIMPs, because the reason they haven't been found yet is probably either they don't exist and the whole idea is wrong, but let's not talk about that anymore, or they're very low mass and very small cross-section for interacting with normal matter, lower than has been limited already. So there's a couple needs you will need in a detector of the future. One is directional detection capability, meaning the ability to see in which direction the WIMPs came, to distinguish them, or the candidate particles came, to distinguish them from a leading background, which is neutrinos. Okay, as shown here, the next-gen WIMP searches will be limited by the neutrino floor. This is the WIMP nucleon cross-section, or the limits that have been set. And this is the mass. And the solid lines here show you a series of existing published experiments. And these dash ones, some of these have recently been published. And what's happening is exclusion plots where they're getting better and better sensitivity and finding no WIMPs. Soon they're going to be running into this yellow region, which is coherent neutrino scattering. A real phenomenon, a real standard model, quite interesting phenomenon, in which, loose analogy to the Mossbauer effect, you have a collective interactions of low, large wave number, low momentum neutrinos with multiple nucleons that exist within nuclei, right? And they can have rather large cross sections, even though the kinematics, because their large wave number isn't as large as other things, other kinds of neutrino interactions. But they're going to be there. And you'll start seeing signals. As these guys get more sensitive, they will start seeing signals. But are they WIMPs or are they neutrinos? You have to be able to distinguish the two. And we know that neutrinos, we believe, and all the evidence is that neutrinos come from a combination of sources. One primary one is the sun, also some cosmological neutrinos and atmospheric neutrinos. So you might, to first approximation, an isotropic background plus the sun for neutrinos. Whereas WIMPs, in the standard WIMP model, disproportionately come from a direction known as the WIMP halo wind. The WIMP halo wind. That's the solar system moving, the galaxy, solar system, the solar system's moving at about 200 kilometers per second through the galaxy. WIMPs are thought to be, to first approximation, at an ensemble average, roughly stationary in the galaxy. They have local random velocities, individual WIMPs like air molecules, but we're running through them. Our solar system is running through this WIMP halo wind. So if you have a detector and you're going through this wind of WIMPs, and there's also neutrinos coming in, and you can tell the source from here different from the source over here, reliably, you might be able to tell WIMPs from neutrinos. Now, if you only, because you know where the sun is, et cetera. If you only know isotropic plus sun plus WIMP halo wind and distinguish those things. If in the end you get a lot of events and they can be completely described by where we know neutrinos are coming from, you've got a nice neutrino detector and maybe no WIMPs. Okay? So that's the idea. It's not a new idea to need directional detection. Back in the 80s, David Spergel at Princeton and others were talking about it. The other need is, like the detectors already have, is to have a large amount of stuff, like the liquid xenon detectors, because you've got, the cross sections are so low, you've got to have a large amount of stuff. So a detector with a large mass. And it helps also to have low Z in the mass. The interactions are expected to be larger with low Z masses, given the kinematics. To date, there have been people doing directional detection with gas-based detectors. They don't have large mass, are not scalable to large mass. Or, of course, with liquid xenon and other things, large mass, but it's a liquid and no directional detection. When there's a detection in liquid xenon, if there is, or there would be with neutrinos, you get a flash of scintillation light. You can see an event occurred, but then there's no scar, no indication of which way the WIMP or neutrino came in. So our idea is to make something like a bubble chamber that can leave tracks in diamond. All right? A cubic meter. Yes, and let that roll in. A cubic meter of diamond with high NV density inside of it. It does not have to be one giant cubic meter of diamond. You need the cubic meter to get enough mass, right? It can be broken up into many millimeter scale sections, either slices shown in this simplistic cartoon or little smaller sections. Diamond is known to be a very good scintillation material. That means when and if there are interactions with WIMPs, and certainly in neutrinos, it's well known how to use diamond and it will create very good scintillation light, and you could localize with traditional detection the event from whatever source to, let's say, a cubic millimeter. Then you'd pull out that section and you'd use many NVs inside to determine the direction of the damage track. And we've done calculations as to what kind of damage track would be created, and it's something like what's shown here in this cartoon. Let me show a slightly more realistic version of the calculation. You have this, the WIMP comes in, or neutrino. It's going to, with low probability, hit one carbon nucleus. That should say nucleus, not nucleon, excuse me. After that point, after that impulse of momentum and energy is given to that first nucleus, forget about the WIMP and neutrino. You then have well understood physics in which that nucleus is going to be dislodged slightly, will hit into the next one, the next one, the next one, and you'll have a rattling damage track within the diamond that will go 50 to 100 nanometers long. And this is many runs. The blue dots are one particular simulation of what happens to different nuclei inside the diamond, and the reddish reddish color there shows you averaged over many events. And so typically what you're going to do is cause damage inside the diamond, right? And you calculate what the strain effect would be on many NVs, shown in black here, randomly distributed at a density that we already can create, and the magnitude of the strain inside the diamond is actually easily detectable with NVs. All right? So that's the idea. The challenges are many. We've done strain imaging with that sensitivity. We've done nanoscale magnetic sensing with that sensitivity, at that spatial resolution. We haven't yet done 3D strain mapping on the length scales and with the sensitivity we need, but I don't think that's going to be the problem. I think identifying and reconstructing the particle direction, if you had enough information, would be fine. I think you're going to have a series of problems like, one, finding the little small region, 100 nanometer region, that potentially has strain in it inside a millimeter region is going to be a problem. So we're thinking about ways to scale that problem and make that easier. All right? We might not just use NV sensing. We might do X-ray or cathodoluminescence measurements to localize this kind of needle in a haystack problem more, right? You're going to have to control the strain background in your diamond and understand this, because you're looking for an added defect on top of what might be there naturally. So we have to work on making the diamond uniform enough. We've done a lot of work on that, but more is needed. And you have to scale everything up. Maybe this works on 1 little cubic millimeter with heroic efforts. Maybe we can inject something which makes a strain defect and find that little thing inside of there. But then to scale it up, if you have cubic millimeter sections inside a cubic meter diamond, can any of you tell me how many sections that is, approximately? 10 to the 9. That's a lot. Does that make sense? Or maybe it's impossible to make work. Maybe we have to find another material that's cheaper than diamond, like silicon or silicon carbide. So we're also thinking about whether we can do this in other materials. I will say, however, the reason we got funding for this from the DOE, not to do the whole big thing, because that's a multi-$100-million project, and underground and all the rest, a pilot study, like $500,000 to do some pilot studies, is because they're kind of desperate in that field to come up with ideas that might have legs, right? And there's also a quantum buzz thing going on now where anything that's quantum seems like today's magic sauce, right? So we'll see. Okay. The interest in NV diamond, hopefully I've communicated to you. I'm trying to be honest. I think some of the applications are really compelling and are already working and translating in other fields and being commercialized and really good stuff. Other ones have got a long way to go. Maybe they're going to work, and some are kind of science fiction-y, like trying to do directional detection of dark matter. I've only scratched the surface. There are a lot of things going on by a lot of groups, and I think it's been a lot of fun, and I see a lot of promise moving forward as well. And I'll say thank you to you, and thank you to my collaborators, and also my group members and the people who support us. Thank you very much. And before I answer questions, though, I'll answer one question that I'll pose that people often say, because when they hear diamonds, they think expensive. And I made it sound a little expensive, too, when we're talking about scaling up to cubic meters. As some of you might realize, well, think about it. Is diamonds expensive? I know diamond rings and jewelry, they're very expensive, right? Have you ever wondered why a diamond ring that you might want to buy for somebody close to you, if you scrimp and save, and the average working person can just make it go? Do you think maybe that's just a happy accident, or could there be a cartel that's designed the prices to maximally extract money from people in return for a fantasy that they've created? Which of those do you think might be true? Right, this latter one. Oh, yes. So in fact, the laboratory-grown diamonds, in reality, which can have purity, they can be cut into gemstones and look as beautiful, because they're essentially identical to the ones that are pulled out of the ground, often in ways that exploit people in the third world. Those lab-grown diamonds can be much less expensive. depending on what you're asking for, if it's a standard one that they're making, not the quantum fancy ones yet, they might be on the order of, for something that's like these little microscope slides, $10, $50, something like that. And if the quantum fancy ones, because they're doing R&D, can get more expensive because we're asking for special features. But if you really wanted to think about economies of scale and have them scale up and make them en masse, it could probably become quite inexpensive. Right? It's really limited by the technology. Once it's stable, it's limited by the cost of power, electrical power that runs the CVD reactors in which they grow the diamond, and the amount of methane and other gas they need to go into those reactors, and a small number of people to supervise the growth. So I answered my own question. Yes, sir.
How long do the damaged tracks last? Do they heal themselves?
Right. So the damaged tracks should not ever go away unless you anneal the diamond at high temperature. I should have emphasized that. But any kind of corrupted feature inside the diamond lattice is going to be thermodynamically stable forever, essentially, unless you take it up around 1,000 degrees C. And so unless you accidentally put your diamonds at 1,000 degrees C and don't know it, they should last. There's lots of challenges here. The logistics of how long will it take us to find, imagine we have a technology which can find this damaged track reliably. But you might imagine it's a bit of a needle in a haystack problem there. That's going to take some time. Meanwhile, the detector, is it going to get another WIMP or neutrino? Well, maybe you have another diamond piece that's known to be clean that you put back in, this sort of thing. How you work out these logistics, what the other backgrounds really are other than neutrinos. There can be radioactive and other radioactive decay within diamond of very low concentration, carbon-14 and 15 and things like this. And other kind of backgrounds. So thinking that through will take time. We're starting it now. But first we want to understand the strain environment better within diamond before you add these damaged tracks, and our ability to make the strain as uniform and clean as possible. Because independent of the dark matter experiment, that is useful to our other experiments. So I wouldn't take a grant and stick a graduate student working on a hopeless or small hope project like this unless it was also useful to other projects and really cool, right? So, and the student wanted to do it. Yes?
So if you take one diamond and you compare different NV centers that you may have in one piece of diamond,
Yep.
and you try to make it as strain-free as you can, how similar are these NV centers?
You want to do like, to what degree is there very natural variation between NVs, even when you try to make there not be? Mm-hmm. Yeah, so that's a very interesting question. So I would say the kind of zero field splitting variation between NVs, when you do your best to make things as uniform as possible, will still be on the order of kilohertz on top of gigahertz, maybe more, maybe 10 kilohertz. Thinking, yeah, 10 kilohertz. I'll give you that number so it's part in 10 to the 5 kind of variation, something like that. Can we do better? Can we get it down to kind of hertz, 10 hertz, 100 hertz? Maybe. So when we think about the people in the community, like myself, and Dima Budker is another one who comes from the AMO precision measurement world. I've worked a lot on fundamental symmetry tests in the past. Dark matter detection is a different thing, but I've worked on that sort of thing, so has Dima. Myself and him and other people have said, to what degree could we use these tools for fundamental physics? Not condensed matter, that's cool, not bio, not geo, not this stuff, but really fundamental physics tests. Like we've done with atomic clocks. And that lies behind some of your question, right? And so they're using them as variations in things like probing damage tracks, or if there's an axion wind that you might see some variation in some field. So you're not actually asking for NV to NV uniformity, but you're looking for time varying things, maybe. But I'm not super optimistic about NVs or other defects in solids as being competitive with real atoms or molecules for ultimate clock or clock-like measurements. Because there are interactions within the crystal, which are real, and though I skate over them and make them sound like it's not a big deal, they're there. And so these are only atom-like systems. They really have differences. Oh, yes.
Is there an existing literature on damaged tracks in diamonds from things like extremely energetic cosmic rays?
Good questions. One of the things we're looking into, and I don't want to answer until we've finished that, I know there are people also in diamond and other materials thinking about leveraging, for dark matter and other kinds of detection, leveraging geological timescales, meaning looking at materials that have sat around on the Earth for very long periods of time, and then examining them to see if they have certain patterns inside them which could only be explained by, it could be WIMPs or other things like that. I'm not an expert in that, but I know there are people thinking about that sort of thing. But that's a good question, and the cosmic rays are typically screened out very well by being deep under Earth, but it's something we have to think about. Certainly in our lab demo experiments that we're going to work on now. Students, questions for students? There's one, you're a student.
There was proposal years ago about buying the concentrated diamond as qubits.
I'm sorry, I can't hear you.
There was years ago there was proposal using the concentrated diamond as quantum bit for quantum computing because of the non-coherence time of the electrons.
Yeah.
Electron spin.
Right.
I wonder if there's any progress in terms of that.
Right. So whether it's NVs or the many other defects which exist within diamond and other large band gap semiconductors, like silicon vacancies, which have an inversion symmetry different from NVs, so they have different sensitivities to things, or defects that exist in silicon carbide, you're right that many people for many years have been thinking about these solid-state spin systems, often optically addressable, as little building blocks for quantum machines, and not just sensors, but actual information processors. I'm not saying the word computing, but you know, it could be that too. And then the question is, what's the progress? The progress is not that great along that specific direction, though there is enthusiasm and momentum now to resume it. And the reason that it has not been great along that specific direction is that the things that you'd like are uniform, strong coupling between many qubits. Let's say NVs or silicon vacancies. That would mean in your mind you quickly have an array. We'll make an array, a 2D, 3D, 1D array, like the people in atomic and AMO and lattices can do, or the ion trap people can do, or the superconducting qubit people can do by having kind of macroscopic things that they can mechanically assemble to have strong uniform coupling. One cannot make NVs or any of these other defects in regular arrays yet. You can control the average density, but then you have Gaussian processes that are controlling things. So if you say, I want a diamond with an average density of, insert a number that can be over a wide range, and I'd like it fully within filling up the whole diamond or only near the surface and that sort of thing, that we can do, and there's lots of progress, as I was talking about here. If you say, I want two NVs next to each other, 5 nanometers apart, so that I can do some fun things with a little 4-qubit system, the 2 electron spins and the 2 nuclear spins associated with the NVs, and maybe I luck out and there's another carbon-13 or 2 nearby and I can get up to 4 or 6 qubits, and I'm willing to search around inside my diamond and eventually find and luck out and find a couple NVs next to each other and write a phys rev letter paper about that playground. Can you do that sort of thing? Yes, you can, and I've done that, and even others have done that sort of thing. But if you want something scalable, where it's not just a few NVs, but it's 20 by 20 or 100 by 100 or whatever, and they have to be 5 nanometers plus or minus 0.2 nanometers or something like that variation, because you want the coupling to be uniform. And we don't want other corrupting spins, other nitrogen spins or other sorts of things nearby, so you can be analogous to ion traps or whatever, but better because it's room temperature solid. There has not been a lot of progress in that. However, the enthusiasm now with the big National Quantum Initiative, there's going to be a lot of money coming for investment in a lot of things quantum, and I'm part of one proposal now that's like some $25 million proposal to make a quantum foundry directed at that exact kind of problem. And there are ideas. This isn't just take a bunch of money and dump the diamonds with dollar bills and somehow it'll work. There are actual ideas based upon things like if you can locally create the right kind of stresses, either with physical, mechanical force, inducing strain or ion or electron beams, you can create higher probability of channeling of the nitrogen, the N2 ions that you'd inject to place the NVs there and reduce the straggle from, if you, instead of 10 nanometers deep plus or minus 5 and a lateral 5 nanometer straggle, get it down to kind of 1 nanometer scale. And then we might be able to get 5 nanometer by 5 nanometer with plus or minus 1 variation. And that's not the ultimate thing, but it's a major stepping stone forward. In the limit of not controlling, only being able to control average properties, NVs at high density on the order of 5 nanometer separation in diamonds that are so dense with NVs they get black looking was used by my colleague Misha Lukinen's group a year or so ago to make these time crystal ideas, these ones where you have subharmonic response with driving that depends upon having disorder in the system and depends upon strong interactions between them. Ion traps were also used. And so there's a whole DARPA program, which I'm part of, and others are part of too, to try to use these kind of new flow-K states of matter where you can basically engineer new states of matter in driven ways, including NVs, and then use them as resources for quantum information. There's a lot that can be done already, that we've still got a long way to go. I think it's wine and cheese time. Do you agree? Yeah.
I think that's good.
Thank you.